My experiment involves the simultaneous inactivation of multiple galactosyl transferases, or GALT enzymes, to create an octuple Arabidopsis thaliana mutant with 8 inactivated genes. GALT genes are responsible for adding the first sugar to the core of arabinogalactan proteins, or AGPs, which are important reproductive proteins. AGPs have many agricultural, pharmaceutical, and industrial applications and are very important in our daily lives. They possess adhesive and emulsification properties and have been found to be effective for immune stimulation, natural prebiotics, drug delivery, and the treatment of diabetes and cancer. This research will allow us to better understand the mechanisms of AGPs and their potential utilizations. This information can be used to genetically engineer plants with altered AGP sugar compositions to increase yield, enhance biomass production, create disease-resistant plants, and more. As the human population continues to grow exponentially, understanding how to increase the reproductive yields of plants through the manipulation of AGPs will become vitally important. AGPs are well known for their implications in reproduction, but the involved mechanisms are poorly understood. These proteins are composed of about 90% sugar, and they are found in a plant's cell walls and cellular secretions. Since the first galactose sugar provides a substrate for subsequent sugar additions, the study will allow me to determine the functional contributions of sugars to AGPs to the phenotypic characterization of octuple mutants when compared to wild-type or control plants. The octuple mutant was created using an existing mutant in our lab, which was created using tDNA insertion methods and had 5 GALT genes removed. We then transformed these quintuple mutants through the utilization of the CRISPR-Cas9 gene editing system, which is a novel technique used to edit the genomes of living organisms. We repeatedly extracted DNA from the offspring of these plants, then conducted PCR, and finally sent them in for sequencing. This information was then compared to wild-type plants to determine how many GALT genes had been inactivated. Once we had obtained octuple mutants, we compared them to wild-type to determine the phenotypic differences between them. The octuple mutants experience visible reductions in silique or seed pod size, seed yield, pollen size and viability, stamen length, anther size, and overall plant height. We determined that the male gametophytes were responsible for the large number of genetic defects in these mutants. Self-reproduction was difficult due to reduced stamen length, which prevented pollen from reaching and successfully pollinating the stigma. We used Alexander staining to compare pollen viability between wild-type and octuple mutants and found that the mutants contained increased levels of aborted pollen and less pollen in general. As you can see in this figure, this method of staining makes viable pollen appear pink and aborted pollen appear green. SEM microscopy also showed that octuple mutants possessed abnormal pollen compared to wild-type.